First off, thanks, Peter, for inviting me for this. So my name is Levi Naden. I'm a software scientist at the Molecular Sciences Software Institute, MOLSI. And we've started a project in tandem with the BioExcel group out of the other side of the pond, if you will, to provide a need that's quickly growing in the community for all of this COVID-19, the SARS-CoV-2 research that's going on in computational modeling and also bioassay modeling, but basically molecular modeling. There seems to be a need that's arisen that we're trying to fill, and there's a subdomain of that of how do we handle all the data that we're getting with that as people are doing molecular dynamics and molecular mechanics simulations. So just to give a quick background of what MOLSI is that we are an NSF-funded institute, and we are created to designed to serve and enhance the software development community in the broad CMS field. Mainly, we're wanting to provide software experience and infrastructure, provide education and training, and provide community engagement and leadership. Basically, filling the role that bridges the gap between having a bunch of grad students and postdocs work in a lab that are transient researchers, to have a more grounded, permanent institute that handles a bunch of back-end infrastructure things, that handles all the training for how do you make good software scientists and good software researchers overall. And we're sort of built to fulfill that goal, so we can actually be long-lasting and serve all the different PIs and the industry partners we might have along the way, and then help bridge those gaps. So what I'm specifically going to be talking about today is our efforts into this COVID-19 pandemic. And the need for this rises because there's a bunch of current ups and downs to the current level of scientific research going on. First off, there's the enemy, which is, of course, this virus particle. And I'm sure you've all seen this around the block a few times. And there's a lot of good things happening in the scientific world. Like, this is an article from the New York Times. I think this was last week at some point, or a couple of weeks ago. But effectively, all of the science and research in the world is being mostly driven towards this pandemic and how do we solve it. Because we happen to have the advantage that this particular pandemic isn't so lethal or debilitating at a global scale that everyone can keep doing research onto it without being completely isolated. However, this is causing a couple of interesting problems. So this is a site that's called the COVID-19 Primer, which scrapes as much as it can from the scientific and news community to track how many papers and publications and how many articles are being written about this virus at the current time. And this has been tracked since mid-February. And as of yesterday, there are over 2,700 papers currently written about this, which is a massive surge relative to how many other papers in all the other fields happen to be. There's a secondary problem, is that the various lockdowns that have gone into effect means we have a whole bunch of really antsy scientists sitting around wanting to help, but they don't quite know how. And so the few things we've been hearing a lot of from our collaborators and seeing all this work being done is that there's a lot of mixed problems that have come up. First off is that because every institute and every lab that under the sun is doing research into this virus and somehow, and because so many papers are coming out, we're getting a lot of weird effects. Like we've heard from at MOLSI through our board of directors and all our PIs that we're in contact with, and then all the editors of the journals who those PIs know, is that they're getting overwhelmed. They cannot keep up with the number of submissions that are going in right now to the journals. And there's a problem in that data is being generated so fast and it can't be reviewed that we're getting a weird mixed bag of scientific results. So I have a tweet here from Tristan Kroll, who does a lot of good work with structural biology here. And he's looking at some of these data that are coming in, and weird things like mismodeled zinc sites that create a weird, what he calls it, a trisulfide, even though that doesn't make any sort of biological sense. And so we're getting a lot of structures coming out of these virus particles that just aren't quality, and there's nobody to really review it. The other problem we're having, and we're hearing this from a bunch of our collaborators, is They've reached out to us saying, we doing research into this, have expertise in this modeling field or do the simulation field. We're getting a lot of requests for people who want our data to do things with it. And that goes back to the antsy scientist problem. And so they're not sure how to get the data to people because, as I'll allude to, a lot of this data, especially in the simulation space, which is what I'll be focusing on, has problems getting around between all the different labs, and it becomes really untenable to do point-to-point transfer of this data. And so our project at Mulsi and with BioExcel spun from how do we answer these three problems right away? How do we get data to people as quickly as we can? How do we do some sort of quality control of that data? And how do we help alleviate the burden of the editors by pre-quality controlling the data, and we can then get it out and get better papers out of this. The other problem we have is that a lot of the good data is really nebulous, and it's unclear how to best translate that. So everyone has their own domain of expertise, and they're great at it, except it's really hard for somebody outside that domain to translate. So here are two different GitHubs that I've particularly pulled. One is the Andrea Thorn lab, and the other is the Salzer lab, who are both doing fantastic work with homology modeling, and then also making a whole bunch of different structure calculations. And these are the results of their GitHub products. At a glance, I can't tell you exactly what's in there. But I can tell you what's in those. I can't tell you the quality of those. I can't tell you the description of those. And somebody who's looking for, I want data to consume and be useful, is going to have to wade through massive folder structures like this. Like the homology model folders have hundreds of entries all labeled this way. And so we need people to be able to come up with short descriptors about these things. And that's part of what we're trying to do. The other problem we happen to have of why the data is nebulous is there's a fair amount of slicing of the data necessary. So in the whole viral pathway of the whole virus infection, if you can disrupt any part of that, from the viral to the host side of things and to the body's response to it, you can stop this virus. But there's a lot that has to do. And I don't know how many people here are familiar with biology and specifically molecular modeling, but just a brief cap of why there's so much data we have to be able to look at here is, so this is the 3D rendering of this viral particle, the whole viral. But if you look at this one of the little spikes, one of the little red knobs on this, its structure at the molecular level looks like this. And this is a protein ribbon structure of this spike protein. And the ribbons are representing different chains of amino acids and everything along the way. And this spike, when it gets down into the host, or gets to the host, binds to a dimer that looks like this. And this is just the spike attaching to the dimer. So there's another large molecule that becomes hard to model, and we don't have the greatest data. We have great data on this one here on the right, and we're still getting better data for the one on the left, which is the spike. So the one on the right is like the enzyme that's in the host membrane. It's in the host membranes of your cellular structures. And so this is then buried inside a massive membrane structure, and pardon the crude overlapping here. And so all of this has to happen along the way, and all of this has to be modeled and simulated and estimated to actually do properties. So there's so much in this viral life cycle that you could attack, that anyone focusing on any one thing is going to miss or not be able to properly describe everything happening everywhere else along the pipeline. And so what we really need is a hub for the easily conveyed science. And specifically, we want people to have science, donate to the hub with simple descriptors that the community can peer review, and then also have centralized pointers or data, centralized pointers to the data that exists that come out of this. Now, a lot of the data that come out of this are all the models and whatnot that people generate, and we are working with the community to come up with the descriptors and coming up with the peer review process for that data. But the big problem is then once you get that data, once you have these models and you start doing things with them, I'm specifically focusing on the dynamic side of things. So any docking algorithm, any virtual screening library, any sort of simulation with this data any dynamics modeling. You run into a problem at this stage is that the centralizer pointers to the data becomes it's a data management problem. And that's what we're sort of that's something we don't have good solutions to in the entire molecular dynamics field. And the big problem with that, and specifically in the molecular dynamics side, is that biomolecular simulations are deceptively large. So what you're seeing here is a single long trajectory of one of the viral proteins that are associated. This is from the D.E. Shaw research group, who are the guys who created the Anton supercomputers, specialized CPUs to do these types of dynamics calculations. And I want you to pay very close attention to this number here in the top left corner. This is the duration of the simulation if you were to translate this into real time. So this particular simulation runs for 100 microseconds. Now, the order of magnitude of the simulations we usually run in biomolecular simulations is on the order of nanoseconds, tens of nanoseconds. And only with really, really powerful computers can you get these really long single trajectory simulations without doing any sort of additional statistical sampling to do what they call enhanced or accelerated sampling. And this trajectory in its full form with all the atoms in the system comes out to be 210 gigabytes for this one trajectory. Now, 210 gigabytes isn't that much data in the grand scheme of things. It's not that hard to do, but this is one group and one simulation of one protein. So the problem becomes combinatorial when you start doing anything else. So if we were to not look at these really long simulations and we look at what other people are doing, we can get some quick estimates of what sort of data people are expecting to have transferred around between the groups. So if we take some of my old work, and this is just a snapshot of a file tree I was working on, these are 12 different simulations, 13 different simulations that I ran, each one for 5 nanoseconds of varying atoms that are in the system, which is why they have different file sizes. And so each of these was just a 5 nanosecond simulation run using a type of additional statistical sampling. So it's not single long trajectory, it's a little more complicated than that, but so it's multiple replicas. And that's why the file sizes get up to this size. So any one group might be running an order of 10 to 100 simulations per biological target. If I were just to look at the specific potential biological targets of the SARS-CoV-2 viral particle, you can break that down into roughly 24 biological domains you could individually simulate. These are things like the proteases. These are things like the non-structured proteins. This is the spike and its derivatives along the way. And there are some people actually doing this work, and we actually have concrete data on how much they're doing. So the Folding at Home project, which is working on this, is a similar approach to doing the long trajectories, but instead of doing a single super powerful computer, it's relying on everyone's home computer. So it's very similar. The name is inspired from what was SETI at home. It's just doing protein folding in the same context. And they're running 46 different structures, and the person in charge of that's estimating there's going to be 10 terabytes of just these simulations, and that's before any analysis, and that's before any domain composition or Markov state modeling that will just make that the size requirement grow. And then if you consider how many potential drug, vaccines, and inhibitors people could develop for any one of these targets along the way, it just becomes a combinatorial problem. So if we were to look at a specific concrete example of what somebody's doing, and actually just instead of many, we'll put one up. There's a project running right now called the COVID-19 Moonshot Project. And that's run by these lovely folks, being headed by Dr. Alpha Lee at Cambridge, and then Professor Frank von Feld, Dr. Artyom Cherkovs, and Dr. Near London, and John Kadera. They're all running this Moonshot Project where the community, people, can submit potential drugs. And they submit structures of these drugs, so long as they are chemically valid. This group is funding through Diamond MX and XChem to make those drugs and actually test them. And they're targeting a single domain of this viral particle, and they've received 3,700 compounds for this one domain. So if you even take the lower bound of all this, where you have one simulation and one group is only doing one simulation for, you see 1 gigabyte per simulation, one simulation per group. On this one domain of just these compounds, you have 33.7 terabytes. And that's the lower bound per group, per domain that we can probably expect. And so it just becomes untenable for us to pass that information around, because we get a lot of, and this gets into some of the loose technical specs, we get a lot of heterogeneous data because not everyone's going to use the same algorithms or techniques. And so we're expecting on the order of in number, like tens of files, hundreds of outputs, thousands of analysis files possibly, that all come out to be, you know, pretty graphs that can go into a publication. And most people, when they say we want these data, they want like the input structures or necessarily the input files, because not everyone's an expert in running the simulations, or how do we set up the simulations? And also because how do we, what are the scripts I need to go from input structure to proper simulation being run through? There's less necessary information that's still useful to preserve, and that's all the provenance information, so logs, some of the more inputs, obtuse input scripts, all the hardware information, all the runtime information, that sort of metadata. And unfortunately, the way people currently do this is they individually reach out to the other groups. And what we're trying to do with this project for Mulsi and BioXL is make it so people can say, here are the pointers to our data with some descriptors and some metadata, and this is how you come to get it. And so the major roadblocks we're currently running into is that groups don't really have, most PIs, most individual labs don't really have the means to maintain large chunks of data, like these large simulation files over multiple simulations, over multiple projects and researchers and domains that you could possibly cycle through. They don't really have the means to maintain that data in a hosted form. Like existing on somebody's spinny box in the drive as a backup server is one solution, but that's not a good way to host data. And then it's harder still for multiple groups with multiple research to share this data in a way that's still not point-to-point contact. And so what we've got so far for this project, as far as what we need, what we have space, is we're working on 2 different sites, or 2 prototype sites. One of them was actually rejected a couple hours ago, so I added that bit there. So we're working on developing 1 prototype. We've allocated a few terabytes of storage locally that Mulsi is maintaining, and then we have proper server storage through Virginia Tech's supercomputer center, like on-campus supercomputer center that we can allocate for this. And so what we're really looking for is how do we, what sort of solutions can we work through together, not just necessarily for this project, but also how do we do this type of data sharing with this very heterogeneous, very combinatorially explosive data for longer terms and projects beyond just this one, because this is a problem that the MD and the molecular mechanics world hasn't solved to date, despite its 50 years of existence. And so this is something that maybe we can talk about, but for now, the main focus is this project. But if we do this right, we can extend this to more projects in the future and also sort of set the way of how do we share this type of data. And so that's the end of what I've got to talk. We have the other 2 software scientists working on this with me are Sam Ellis and Andrew Abi Mansour. And then also have to thank the people running this project through BioXL and Mulsi as well, which is Teresa Head-Gordon, Romy Amaro, Eric Lindahl, Rosanna Apostolov, John Cadero, Rafael and Alex, who have all been helpful in gathering up some of this data and helping explain, helping me figure out all the different biological domains, as I'm not a particularly good biologist myself. So they've been helping a lot with this biological science side. And then thank you all for listening, and I'm happy to be a part of any discussion you want to have.
Excellent. This was a very useful talk. Thank you, Levi. So I could offer some.
Give the word to Mario.
So I will leave it open. Who wants to jump in?
No, I was saying this half jokingly. I mean, it's. Our community, of course, does have management systems that are geared up for the multitude of data sets you are talking about and so on. Where we are weak on our potential data privacy and data protection issues, because our data doesn't have the multitude of organizations that might have individual rights. But in principle, I mean, what we use the WLCG and we use our the scientific computing sites, and we do have systems that where other communities are already looking into, which we can probably offer. Leave it here for the others to come.
And perhaps to add to this, we have also integrated our data management system with Boeing as well, because we're running the Atlas at home. So we do have knowledge in dealing with these volunteer computing systems as well. For privacy and data protection, right now we mostly depend on having a, let's say, safe infrastructure. So this is definitely something we would need to look into.
But maybe we can ask Levi, are there really, for these cases, are there any privacy issues, or this is all open data that you're talking about, right?
So in theory, all the data is open. Not all the data. In theory, most of the data is open. There are the cases where it's proprietary research, proprietary simulations, partnerships with industries that don't necessarily want to share what they're looking at. And in some cases, people will be reluctant to share their results before, say, publication out of fear of being scooped or something, or because of other legal issues. But for the most part, most of these simulations people run and work on, and especially what people are working on for the SARS-CoV-2 virus, that's pretty much all in the public domain out of need, and then licensed as gently as possible, and as permissively as possible. So it's pretty, I think the problem is that there isn't going to, the shorter answer is there's not really going to be a problem with licensing. And if there is, they just won't upload, people won't just won't upload the data. Simple as that.
Mm-hmm.
So the thing that we had, maybe had already been discussed a little bit with the PI of MolC and others is that, as Marcus was saying, is that, there is this open data repository, Zenodo, which, CERN has put, I think, 100 terabytes behind. To put some, there's not, the scale of the data we're dealing with is, hundreds of petabytes, exabyte scale. So in terms of the repository, there's something there that can just basically be used today. The metadata parts that you're talking about is obviously not something that is built into what we're doing, but it's built into what you're doing, sounds like.
Right, and the idea is basically we need a way to share this data with people generally, and that would be a useful solution. And then at least for this first project, is we track the metadata and just provide pointers to where it's being hosted. Yeah,
and I think we have different approaches. Zenodo is something, is a single point of entry, but it doesn't have a data distribution system to compute sites at your university, where you can just, I mean, if we're talking about in 20 gigabytes or so, you can probably download this. But if you're talking about many terabytes, then that becomes not always practical to do by hand. And you want to have automatic systems, and there is an option for putting something behind Zenodo which would do this. But that immediately brings us to the question is, where are your compute sites? And I mean, are you linked to infrastructures like the WLCG or like the OSG or in any other funding body where we can basically then install such systems for data management and data distribution?
I'd have to look into that for MolC specifically of what we're linked into, but the problem we're going to have is that all the people who will be coming to this to gather data to then run their own simulations aren't necessarily, they're not centralized. So even though we MolC are at Virginia Tech, we have, like, when I say collaborators, I mean, we're talking global scale need and want to participate in gathering data and doing their own work at their own site with their own compute resources, and not necessarily trying to queue up or execute compute resources through other means by just directly accessing the data. And that's partly where the problem is that a lot of these PIs are stationed out of institutes, and those institutes maintain their own compute clusters. and they just run it all on those. And that's a fair amount of the use case, but I couldn't give you a number on how many I think.
Can I just to follow that up,
Yes.
for the folks that are doing that, and you may have said this in my talk, but my home internet crashed, so I missed a chunk. I apologize.
That's all right.
But for the folks who are at the individual universities, what size data sets are they going to want to be running over to run these simulations?
Right. So the amount of data they will likely, and this is where the problem gets in, is that the amount of data that they will likely want to access is for the people doing any sort of modeling or that take the models themselves. We're talking trivial amounts of data. We're a few gigabytes at most, and that's probably pushing it. The output of what those groups produce, however, that can be on the order of the hundreds of gigabytes scale. But then the big problem comes to then how do they get that? How do they then recycle that data back in? So then the people who look at those types of data, who then do like advanced analysis on those. So for instance, like that movie I showed where it's a really long
lost
projector.
the internet again.
The short of it is most people will want like order gigabyte data. Several people will want order hundreds of gigabytes of data. And then the output of those people will be a few hundred meg, maybe. But the problem is there's so many points of people accessing that it just becomes, it's not something that's easy to do.
Have you been engaged with the HPC consortium? I mean, that seems like a bit of a rallying point around a lot of the challenges you mentioned. And Open Science Grid is part of that. And has MOLSI or you engaged with the HPC consortium?
Yes, MOLSI has engaged with the HPC consortium on a few different projects that are not ones that I've been associated with, but it is something we've been engaged with. And we're actually, for this particular project, we're reaching out to about as many people as we can and all the different contacts we can. So this is just, so this conversation is just one conversation of many that are going on in parallel as we speak.
So can I ask, I mean, we mentioned the WLCG and the OSG, but those are entities that sort of originated with the hydrogen physics community, but especially the OSG has a broader mandate that has essentially these kinds of data distribution mechanisms to many institutions. It's essentially, because we have essentially the same problem, how to distribute data at this level and even sort of larger data volumes in a capping sense to many individual institutions. And of course, we're plugged into most of the major research institutions, maybe not some of the medical ones, but in the world. So there's the two parts here. There's the sort of data hosting part, which you were talking about, and then there's the sort of distribution and maybe caching. And we have essentially these structures to most of the major research universities.
Yeah, I mean, we could also, I mean, if the data volumes are not strikingly large. I mean, so that we could argue that a lot of that work requires some data distribution services like Russia FTS and what have you. But it needs a bit of organizational blessing, which we could get by talking to Simona and the WLCG folks, and then within the OSG community, and kickstart something which we can put into effect, and we could even link that up with Zenodo in one way or another. It needs a bit of brainstorming, but I mean, there are a lot of things thinkable which we could do in relatively short times. I mean, the other thing is what Lukács is doing. Is directly giving access to Binder and to mybinder.org for analyzing the data, which is on platforms like Zenodo, which is easy to do. And so instead of downloading it, you would get access to it on cloud resources right away. So I mean that's the other one, and then we have to find for the support for actually financing the cloud resources. But I think at the moment they're basically for free.
So I guess let's turn it into a question to Levi, which is that if, essentially what we're saying is some of the structures you're talking about we have for data volumes and number of institutions like you're talking about is you know, that the part that we of course don't have, that's your expertise, is really sort of managing the metadata and the community engagement on that side. Are these two things that we should be looking to try and continue to try and bring together such that we can provide some of the underlying infrastructure you need?
I think that would actually be the best way to go about this, because this is a problem that has plagued the molecular mechanics and molecular dynamics community for years and years and years, is that we don't have good ways for really long-term storage of data that doesn't involve tons and tons of money to basically buy permanent allocations on their local compute clusters. But then B, how do we distribute the data to researchers? Now, there is the trade-off, there's a caution that we have to be very careful about, and this is partly where we come in, we being Mulsi, is we have to be careful not to turn this into just a dumping ground where people go, oh, here's a whole lot of space we can just throw things onto, because it as I mentioned from the quality part of the talk, is that it's very easy, and there's currently a lot of not so great science going on right now. So it's very easy to actually make a lot of data that is effectively garbage. Like that stack of simulations I showed from my own slides and my own work, that was attempt 3 after doing a whole bunch of different parameter tweaking to see if I got something that was reasonably stable in these simulations. So we just have to be careful about when and how people add data to these resources, these distributed resources.
So as an example, the way we deal with it, we let people create the data, and then we let them annotate which one is actually the good data, and then we just let the old one expire. This makes it, this makes the whole process a bit easier to follow, because sometimes you don't know which data is good until you are some steps down the line in your processing workflow. So we just let them dump it there, and then we see how it moves.
That would actually probably be the simplest approach, and it sounds like you guys have probably thought about this a lot longer than I have, as to how do we handle groups who are just dumping data. And I guess I'm assuming if they never flag or annotate the data as good, it'll all expire.
Yeah,
they are popularity-driven systems. Right? Yes. I mean, we trace how often people access data and these kind of things. I mean, there are many things in the different classes of data. It's a very complicated system. We are paranoid about losing data, and we have a cost problem because we are storing half an exabyte of data. Right.
Yeah. And okay. So this is, you guys have been thinking about this a lot longer than I have. Anyone in my field has. So excellent. Yeah, that sounds like.
I've got a somewhat orthogonal set of questions, and it starts at the big picture level, which is where we have no expertise, none. Is what sort of users would there be? What would they want in terms of an interface? Can you define use cases where people would say, I want to provide this data, and other people say, well, I would like access to this type of data, and can you agree on a common language for how it should be described, labeled, stored, to what extent there should be APIs so that people can use it, written in Python or R, whatever languages you all use. Mm-hmm. Because I think that before anyone starts to design an exact structure for storing data and putting it in files and figuring out the access, multiple people from your community will need to agree roughly on what is needed.
Correct. And that is something we are still working on. So the prototype site that I had mentioned that is where we are going to get the feedback as to what people want and how they want to access this data. Because that is something that's currently, I imagine you've experienced this before, it's very difficult to get that information out of people, especially when you tell them, I need a response within about 48 hours of what you want, and they have their own thing. So that is something I have how I would like to access it, but I don't necessarily agree that that's how the community wants it. So I don't have a good answer for specifically even this data right now. And that's also part of why we the molecular mechanics and molecular dynamics communities haven't come up with a good system yet, is nobody can agree on what they want, because yes, those really long simulation trajectories are interesting and useful at certain extents, but there's so many other fun statistical ways you can run simulations that have wildly different goals and wildly different needs and methodology under which they're trying to accomplish. So I think it's a matter of, we need to get feedback from people on the site first, and then we can figure out how do we prototype that data and then start getting design things from that. And that's why we have this sort of pseudo-temporary space that MolC is providing for people right now to see how are they going to deposit, how are they going to then access it, and where are the friction points.
So it seems to me, just in a very naive way, that it is better to do something than nothing. And then it's the next step would be to plan to revisit in roughly 3 months. That could be 2 months or 6 months, but plan to revisit, trash everything you've done, and start over again, which sounds painful. But I've never known anyone to design any software or anything else well the first time.
I agree with that. And even just to give a concrete, this is the third redesign of the prototype site that I've been doing since I started it 2 weeks ago, based off feedback. So yes, I think that's probably a good approach.
My next question was who you think your partners are, because at the end of the day, meaning for COVID-19, not for your community, not for MolC longer term, which has a much broader academic scope, but for the next 6 to 18 months, if the focus is the SARS-CoV-2 virus, at the end of the pipeline, you want to be working with people who are doing pharmaceutical development. And that's the goal, right?
Yes, that is probably the goal of that is a useful goal for MolC, that is a useful goal for this institute, or this partnership that MolC and BioExcel have together.
I'm saying in the short term, there's a very specific use case, which is why everyone in the world is working on this right now. And it is not for its intellectual interest, even though it's fascinating, but the real goal is to produce something to make the world better on a short term. And so one of the things that you really have to think about very carefully is how do you go beyond the usual academics and engage the scientists from the pharma world to make sure that what you're doing has some possibility, at least, of informing their work.
But I think this came in a bit through I forget what it was called, but the collaboration you described later in the talk, right?
Yes, I think I don't think it's been ignored. It just seems to me that has to be a guiding principle for the short-term project.
Yes, and the way that at least my side of things is approaching it is that MolC will serve as the facilitator for all of the infrastructure and basically the design and providing the resources for people to access this. And then that the individual spin-up groups or individual researchers in this area will be providing the actual science, which then will be conveyed back to the endpoint partners, the pharmaceutical companies. And so there are a lot of these scientists who are wanting to contribute to this, are working themselves already with pharmaceutical companies along the way. Like the COVID-19 moonshot that I mentioned, they're actually in the contacts through Diamond and XChem are tied to pharmaceuticals. So I'm not sure that MolC and BioExcel explicitly are going to be having these partners, but they will likely come out naturally as the people who are contributing and receiving data and giving all this feedback along the way. Those people are the ones who will likely have the notion of what those endpoint pharmaceutical companies want out of this particular type of data. So it should come about as part of the process, but it is definitely something that we're going to be striving to keep in mind as we make our own design and implementation decisions.
So are there other questions?
Yeah, I was just going to follow up really quick. I think Mike brings up a really good point. It's something I was wondering too, and something if we touched on you can think about, is when you say access, do the consumers that are doing the science just need access in the sense of just, getting the data and pulling it locally, or do they benefit from some, cached, some interface that maybe brings them into a Python or R framework? So those are two very different things. I don't know what that community looks for and what they use and what would help broaden the science scope too. What I mean is involve more people.
Right.
So anyway, I think that's an interesting question. I don't know the answer to it, but something
there are two answers to that. Is that in the very, 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 very short term, the things that people need is they need data to work with, and they need to know what of that data that has been generated and deposited is the most helpful. Like which one has the correct structures, which one has the correct biological signals, which one is best for their particular domain of expertise. And that is a parallel thing that we are working on as part of this project, where we are reaching out to all of our scientific contacts. And by we, I mean MolC, every person involved with this, BioExcel, every person involved with that, and all their contacts, and then all the institutes they're aware of, and trying to say, can we get people to agree to do pseudo quick reviews of this data? And then that gives us a way to actually filter this huge array of data that people can access. And then that gives people the data that's higher relevance, higher biological function, says this is the best reviewed data, go with this. And that's their starting point. They will then likely pull all that down locally and run, because those people will have their own resources. And that's part of the beauty of having everyone in the world working on this project or this problem, is that everyone has access to their own resources. They just need a starting point. They need something to work with. And so for now, we don't have plans to have people have access directly to a Python terminal or binder thing that's connected to some high performance computing on the back end as part of this project. We just want them to be able to know what data are there, because so much is being generated, and know what data are quality for their specific domain.
I got it. Thanks. That was an excellent answer. Yeah. So the priority is really about curating and sort of discovering what the most useful data is, and then the two issues around access are secondary.
Correct.
Okay. Thanks.
I've got another super technical but naive question.
Go for it.
When people want to get data sets, will they be part of the regular internet, or will they be part of Internet 2, or both?
So when people get the regular data sets, right now, Internet is fine. We don't need the advantages of Internet 2 for a lot of this stuff, mostly because many people will only be interested in a large number of files that ultimately constitute a small file size, or one or two very specific files that constitute a moderate size. And moderate in this point is like order terabyte at most.
If terabyte, transferring a terabyte of data at ordinary speeds can be painful.
I agree, but I also think that most people in doing the research right now, some will have Internet 2 access. Some will have Internet 2 access. Most people will just have Internet access for now.
Just something in the back of the mind for design is that it might be interesting if this is designed to have some Internet 2 access as well for the larger data sets.
Probably, but I think designing...
Not first order problem, I understand that.
Yeah, but that's sort of where I imagine most people. Like, I imagine most DCPIs have some ability to access Internet 2 protocols and pipes, like through the high performance computing centers or the HPC resources at like their local campuses, or that individual research companies might have access to a pipe, but a large chunk of this will just go over normal Internet protocols.
So here's another question. This is completely different, which is organizational. Is this work that you're doing with one or two other people? Do you have any sort of an organizational structure where people, like an advisory committee?
So this project or the products that people are producing for this project?
This project of providing an interface.
I'm not sure I follow the question.
What Mike is shooting for is something else. I mean, it's if you wanna have a model for data sharing in the community at large, as you were outlining on your slides, there has to be some form of organization behind the community to make this actually work. I mean, because it comes with long term, not short term, not now.
Yes.
But
So
it comes with some sort of the responsibility for funding the compute resources, sharing resources, and these kind of things. And for getting there, you also need some sort of organization that has been put in place, or maybe you have it already, that can actually get you from where you are now to a situation where you want to be, where the system is really up and running in its full glory.
yes, so I imagine that MOLSI as an institute itself would be that driver for that, because we were created by the NSF explicitly to help solve these types of problems, where researchers and people doing all their own work, the nature of they need to focus on their work doesn't let them focus on the infrastructure side of things, and this would definitely be an infrastructure side of things. And so we were created to sort of try to fill those gaps, and that would be, we would probably be the central institute for that, with a few additional partners depending on how the project forms. So I don't have a good sense of if it would be anyone beyond MOLSI at the moment, but it would likely be this institute itself.
Okay, so anyone else want to ask questions beyond those that have? Okay, anyone else that has already been asking questions want to throw in any last questions?
This is Vasil here. I wanted to ask, and what is the data format? How do you store the data?
Take your pick. A lot of the data are stored in, so a lot of these input structures are actually stored in plain text, just usually some sort of formatted plain text, or something that can be at least translated into plain text. And then a lot of these larger data that are like these simulations data are, they're basically glorified n-dimensional arrays with that like are amenable to like H5 formats. They could be cast that way, but usually also have a bunch of additional metadata attached to it that doesn't make sense. There's no uniform or even agreed upon data standard in the molecular mechanics community. I could sit here for the next several hours probably and list file formats I know other people are using, and they'll be exotic or unique to the program which generated it. And then varying levels of compression, not compression, split over multi-files. It's a mess.
Yeah, because I've seen that some of the sequencing machine output is plain text, and it can be quite big.
Yeah, there's an alarming amount of plain text, like not necessarily even human readable, but just plain text files out there. Like all the protein structures, those PDB structures, they're protein data bank structures, those are plain text. Those are fixed column plain text files. A lot of these input coordinates are just plain text files, although in reality they're just arrays of floats. But then all the metadata associated with, well, how does this particle at this point translate into a carbon atom that's surrounded by these other atoms in this particular chemical environment at each frame of the simulation, because that can change depending on what type of simulation and what sort of analysis and statistics you're doing. So there's a bunch of additional metadata that doesn't fit well into just making simple like H5 arrays. So it's a mess.
Okay. And when you were giving numbers on the data, that's uncompressed text files, or this is after some sort of a compression? Do you have an? So
the numbers I gave for like the actual sizes is what is the upper, my loose approximation of the upper limit of the total files that they output, and their default, whether they're compressed or not. So for instance, the movie I showed, that's 200 gigabytes of uncompressed movie data, but it's in a file format that I think starts compressed already.
Okay.
Hi, this is Lucas. I joined late, so maybe this was discussed already, but regarding the data format, so one thing that our community is kind of specializing is these types of hierarchical or more complex data that go beyond like flat, like NumPy arrays that you would normally see in HDF5 files, because our data is pretty nested. You know, we have particles that then decay or something, and I don't know if that's relevant, but it might be. It sounded like there's some kind of connection where like we also are not fine with just like flat arrays.
You know, there's probably, I imagine you guys have solutions, or at least have been thinking about these types of data as well. And if we can generalize them and say, what's the tech necessary? And then get people to go from there, just to give, but I don't know. That's going to be a very hard uphill battle to overcome. There was a conference in November I was at, which basically had the developers from all the major dynamics programs that I can think of. And they were trying, and so one of the goals of that was try to come up with what's a trajectory format, which is the propagation of time of these molecular systems that we can all agree on, like at a meta level, like what sort of metadata, what sort of data need to be captured. And can we start developing a spec for that? And there was not an ingredient that was able to come out of that, at least in something that resulted in a tangible product. Good ideas, but not a good product. So the space has been trying to tackle this, but I think that a lot of the space would be fine with a solution that works, that they can either force or adapt or extend to meet their particular needs. So it's probably shares a lot of overlap. We just need to figure out how to hook those things up.
Does mostly, so this thing about file formats in other communities like crystallography, there was this CCP4 that by defining this, and I've heard the same thing from like the geoscience community that, you know, but is this something that mostly would actually consider as sort of ownership for kind of developing?
This is something that MOLSI has discussed wading into. The problem is that we don't have necessarily, the problem is that it has basically been decided that there are better projects that we can exercise our staff's time on than trying to get the communities, the various sub-communities that have all been fighting about files and file formats and what should go into a file, what shouldn't, for their entire existence. So we haven't waded into that space explicitly, although we are trying to keep an ear out for that and influence it as we can.
In some sense, it could lead on from just organizing the sort of meta data level that you're already focused on, eventually a common repository and things like this.
If we can do that, that'd be great, but it's not something we've explicitly waded into. It's not a space that we're quite ready to wade into yet, just because the individual spaces already have their own infighting going on in there.
Other people, other questions? Maybe there was somebody else. Anybody else wants to come forward with a question? Okay, this was great, Levi. Thank you very much.
Yeah, thank you for inviting me.